Good morning, good afternoon, good evening, everybody. It's Dr. Kaz. I'm bringing our third installment into the digestive system. And I'm going to just kind of pick up right where I left off with our last video here. And we just completed talking about the stomach and specifically the intestinal phase of our digestive process, which brings us to, the lower GI or gastrointestinal tract and all the organs therein. All right, so keep in mind, the process of digestion does require us to break down substances from something that's large into something that's much smaller, and then absorb any nutrients from that. And that's what we're going to do here. We're going to continue to digest in our lower GI tract organs, and we will continue to absorb, much of the molecules and substances during that digestive process here. And as we move through, the ingested materials, closer to the terminal end of your digestive tract, we are going to see, more and more in proportion of that material that's left is going to be undigestible material or unabsorbed material. And so we're going to eventually start to package that up into feces and for elimination, but we'll talk about that more so when we get into the large intestine. So let's start off with the small intestine here. All right, 3 regions to the small intestine: DJI, the duodenum or duodenum, the jejunum, and the ileum. Okay, the duodenum is the immediate part that comes right off of the stomach. So the pylorus and the pyloric sphincter, are going to transmit chyme, from the stomach into the duodenum. Now, the duodenum is actually still considered to be part of the upper GI tract, but we'll talk about it here with the lower GI tract. We briefly talked about it in our last video here. Okay, so during that time, as that material is moving from the stomach, the chyme is moving from the stomach into the duodenum, we're going to start to mix it up. We've already mixed it up with the gastric digestive juices from the stomach, but now we're going to start to mix up that chyme that was received from the stomach with the digestive juices and secretions that come from the small intestine and from some of the accessory organs, namely the liver, gallbladder, and the pancreas here. And so we're going to mix all of that up here in the duodenum here. So really what we're going to see, this is really a crucial area of your digestive system or the GI tract. We're going to see a lot of chemical digestion occurring here, and most of the absorption is also going to occur in the small intestine. All right, so it's really, really crucial. That's why if you have damage to the small intestine or for some reason you've had to remove some of your small intestine, the chemical digestion and absorption is highly compromised in those types of situations. So you've heard me mention the term about accessory organs. Well, we're going to talk about some of the accessory organs, These accessory organs are going to secrete, making some of those digestive juices like bile and pancreatic juice. Know that bile is produced by the liver, but it is stored, and concentrated by the gallbladder. A lot of folks think that the gallbladder makes bile. It does not. The liver makes the bile. That's why if you have your gallbladder removed, you can still produce the bile, but it's pretty much going to be not as concentrated, and so it'll still cause some issues when it comes to digestion. And then, of course, our pancreatic juice and some of the comes from the pancreas. Okay, that pancreatic juice, we'll talk about it, is loaded with these wonderful digestive enzymes, that will help to break down more of that ingested material there. All right, next in the lower GI tract is the large intestine. All right, this is kind of like the cleanup batter. It's going to finish up, with a lot of the absorption for pretty much what has not been absorbed, but we see quite a lot of water being absorbed here in the large intestine. But at the same time, we'll grab onto some remaining electrolytes and some vitamins. And then as we move from the proximal end of the large intestine towards the distal end of the large intestine, we're going to see the liquidy contents become more concentrated and more compacted, and we will produce what we call feces. And we'll eventually get rid of those feces through the anus. right? So we've seen this a little bit before, all right? This picture when we were in chapter 1 in lab, right? You can see here is the duodenum, the first part of your small intestine. And as it's rolling through here, we'll move into the jejunum and then finally the ileum. Then the ileum empties into the first part of your large intestine, all right, through the ileocecal valve. Down here below the ileocecal valve is the cecum. It's a sac-like structure. And then we have our ascending colon, right? And it takes a turn here, crosses the body at the transverse colon, then goes back inferior to the descending colon, and then makes this kind of S pattern. That's the sigmoid colon. And finally, we get to the rectum down in this region here, right, where the feces waits for elimination out through the anus. And then we can see some of our accessory digestive organs here, right? Here's the liver, okay, the pancreas, and then attached to our liver is going to be our nice green friend here, the gallbladder. All right, so let's talk about that initial part of the lower GI, and that is our small intestine, also known as the small bowel. All right, so pretty much the material that you've ingested, and once it leaves your stomach, and the material can reside in the stomach anywhere from 2 to 6 hours, well, guess what? It's going to spend about another 12 hours in the small intestine. So if you're going to the bathroom today, that's going to be pretty much material that you've ingested, right, the previous day or way early in the morning if you're going to the bathroom later on in the evening. All right, so about 12 hours in the small intestine, but keep in mind, right, this is where we're going to see, all right, the most or the largest amount of chemical digestion and absorption is going to occur here. So we're pretty much going to grab onto most of the nutrients that can be absorbed here in the small intestine, right? And then we're still, and I know I mentioned this, that the large intestine plays a role in the absorption of water, electrolytes, and vitamins, okay? But that doesn't mean that the small intestine isn't doing that, right? It's pretty much the large intestine, like I said, is the cleanup batter. It's going to grab onto, right, what the small intestine couldn't get, right? Because we don't want to waste anything. Your body is a great conserver. It likes to hold on to things for a rainy day. So let's look at the small intestine's anatomy. All right, so it is, all right, a tube-like structure. It's going to have the 4 layers there, right? And in here, we are going to see, all right, those 3 segments, the duodenum, the jejunum, and the ileum. So the duodenum is actually going to be made up of 4 subcomponents. You don't need to know those 4 subcomponents or smaller portions there, right? But the duodenum starts at the pyloric sphincter. It's going to be on the distal side of the pyloric sphincter, okay? So that chyme, all right, emerges into the duodenum here. All right, and then the duodenum kind of makes this C-shaped kind of configuration coming off of the stomach, and then it comes back, and then it'll shoot backwards. So it's going to kind of look like a little bit of a C here, and the pancreas sits in this area right here, right? So where the duodenum meets up with the jejunum, we call it duodenal jejunal flexure, right? And you should know this, right? The duodenum, the majority of it is going to be retroperitoneal. That means it's going to sit against the posterior abdominal wall there. It'll be outside of the serosal membrane there, right? The first part, right, which would be, and I know my drawing is not the best, imagine this being the stomach over here, right? So the first part right here, that'll be intraperitoneal, as is the stomach. So in the duodenum, we're going to see a lot of chemicals being dumped into this thing, right? So the chyme, which has all those gastric digestive juices that the stomach produced, all right, and it still has a little bit of saliva, all right, or I shouldn't say saliva, but the digestive juices that was found in the saliva, the salivary amylase and the salivary lipase, okay, all that is mixed in. So now this chyme comes in, and remember that the chyme is going to be acidic. So it gets dumped into the duodenum. So the duodenum then is now going to receive, all right, secretions from the liver, the gallbladder, and the pancreas. So not only is it going to be getting, all right, the chyme that has all the stomach secretions, it's going to have secretions from our accessory digestive glands, all right, the liver, the gallbladder, and the pancreas. So a lot of stuff gets kind of dumped into the duodenum there. All right, the middle part of our small intestine, right, that is the jejunum. Okay, it's about 2/5 of the small intestine. This is, again, where we're going to see, all right, chemical digestion and nutrient absorption. So digestion, absorption on a chemical level, right, is going to be found in the jejunum, and that is going to be the region of the small intestine that does the most when it comes to chemical digestion and absorption. All right, this structure is also intraperitoneal. All right, and then our terminal end, the last part, is the ileum. All right, and the ileum is going to make up 3/5 of the small intestine, so quite a bit here. The ileum will then terminate at the ileocecal valve, and that's the valve that separates the small intestine from the large intestine. And so we have a reflex that we'll talk about here that is going to stimulate the opening of that valve so we can move material from the small intestine into the large intestine here. And this structure is also going to be intraperitoneal. It resides in our serosal membrane there, right, or the peritoneum. All right, and of course, we're going to still absorb, all right, the material that we are chemically digesting here in the ileum. In the ileum, and I'll show you a picture here. I believe it's on the next slide. Yes, it is. All right, the lining of the ileum have these circular folds, all right, and those are called plicae circularis. And so they're made up of the inner two layers, the mucosal and submucosal tunics there. And so the nice thing about, all right, the plicae circularis, these circular folds here, is one, they increase the surface area, but also they act as speed bumps. They're going to slow the movement of chyme, all right? And if you've ever seen anything having to do with glaciers and how they move, I mean, glaciers are a flowing river of ice. And so glaciers will, when they're moving, they kind of churn up the bottommost portion of the glacier itself. And that's what happens here, right? As the chyme rolls across these speed bumps, these plicae circularis, it causes a churning, right? So again, we're increasing the exposure of the chyme to the intestinal wall there. And so we can increase our nutrient absorption, right? And we slow down that movement of chyme, so we give our intestines more time to absorb that material. So as we move, all right, from the proximal portion of the small intestine to the distal end, which is the ileum, right, we start to decrease in number, right? So we're going to see more in the duodenum and the jejunum, and then less in the ileum. Okay, so it just starts to slowly kind of transition to fewer and fewer. So here you can see in our picture the circular folds here, and you can see how they'll act as speed bumps here, but at the same time too, all right, when we're looking at it from the side, it creates a larger surface area here, because now not only are these areas going to be exposed to the chyme, but all the way down in here will be exposed to the chyme, and even the sides here, all right, of the folds. So your body has found a nice way to increase, right, our surface area here for maximal absorption of the material. So in our small intestine, you guessed it, we're going to have, all right, glands, all right? The intestinal glands are going to be found, right, in these structures that we call the intestinal villi. And the intestinal villi are going to be these projections that you'll see, right, on the mucosal surface here of the small intestine. So they'll be lining all along, all right, the intestinal villi. And their job is going to be to secrete, all right, digestive juices, these enzymes, this intestinal juice, all right, for chemical digestion and help maximize absorption. So there's an old friend of ours that we recognize that we've seen before, the goblet cells. All right, the goblet cells, their main job, okay, is to produce mucin. It's a glycoprotein. When mixed with water, it makes our wonderful and much appreciated mucus. Mucus's primary job, okay, is to act as lubricant, right? Because the movement of chyme, okay, it's an abrasive process, so we don't want to damage the epithelium there. So we lay out a lot of mucus to help with that, so we're not causing a lot of damage to the intestines. And that mucin, right, or that mucus will help to buffer the acidity of the contents there of the chyme. So as we go from the proximal end, which is the duodenum, to the distal end of the small intestine, we're going to increase the number of goblet cells. Right, so that's going to be different than what we saw with the plicae circularis, or those circular folds. As we went from the proximal end, right, the duodenum, to the distal end of the small intestine, the ileum, the plicae circularis, the circular folds, decrease in number. Opposite for the goblet cells here. Right, and yes, goblet cells are unicellular glands. Well, we have another set of unicellular glands here, right? And these fellas are going to produce enteropeptidase. Ooh, wait a minute. Let's look at that. What is that peptide? That has something to do with proteins. You're darn right. Okay, enteropeptidase is an enzyme that's going to continue to help to break down the proteins, right? Because proteins are a bear to digest, right? The stomach helped, all right, with the hydrochloric acid, all right, and pepsin, right? That helped to break down, right, proteins into illegal peptides. So now we want to further break down those illegal peptides into smaller peptide chains. Our enteroendocrine cells, okay, endocrine has to do with hormones, right? So of course, we're going to discuss CCK, cholecystokinin, and secretin. We briefly talked about those. Those are two hormones, and we'll talk about them in a little bit more detail in a few moments, right? That are going to they're going to oppose gastric secretions and gastric motility, but they're going to have some other roles here too. And then in our submucosal region here of the small intestine, remember, chyme is acidic, so we're going to produce an alkaline mucus here. And so that's going to protect, all right, the lining of the small intestine, the duodenum, from the chyme. Right, because it's a pretty hostile environment when it gets that dumpage, all right, from the stomach with that low pH, because you get the hydrochloric acid that's being pumped into there, all right, and that has hydrochloric acid has a pH value, all right, close to 1, all right, but when we mix it up with the other contents, your stomach acidity, the pH is usually between 2 and 3. Still, it's not a good thing. All right, so here you can see a nice picture of one of our intestinal villi. All right, so these intestinal villi, in the center of the intestinal villi, you'll see we have a lymphatic structure and we have some blood vessels. Again, all for the absorption of materials that are going to come from the intestinal lumen. So lining, right, the lumen here, right, of our small intestine, we're going to have simple columnar, meaning just one layer, simple columnar, right, non-ciliated simple columnar epithelium. We have microvilli here at the very tip. And now the microvilli are going to also increase the surface area for maximal absorption here. Right, so these cells, their primary job is to absorb nutrients, right, get them to diffuse across, all right, the epithelial layer here and into, all right, the blood vessels and the lymphatic vessel waiting on the other side. Then spread out throughout the intestinal villi, you'll see the goblet cells producing the mucin, which will mix with water, produce mucus. Okay, then we move down here, all right, down at the bottom, you're looking here at our intestinal gland, way here at the bottom, right? We have one of our unicellular glands, and this is the one that helps to make that enzyme, the enteropeptidase, that is going to help with protein digestion. And then way at the bottom of that gland there, of the intestinal gland, you'll see the enteroendocrine cell, and that's going to be the cell group that secretes, all right, CCK and secretin. All right, so We've talked about it in the stomach. We've got to talk about it here in our small intestine: movement, motility. Right? We saw how the stomach, all right, there were two processes for motility in the stomach. We had mixing, the gastric mixing, and then the gastric emptying. Right? Well, we're going to see, right, the same thing occur here. We're going to have mixing here in the small intestine, and then we're going to have movement or propulsion, right, of the ingested materials, right, from the proximal end of the small intestine to the distal end. So of course, the mixing, okay, like we saw in the stomach, we're going to now mix the chyme with the glandular secretions from the duodenum, right? So we call that segmentation. And it's just like taking a glass, you pick up a glass that has very little fluid in it, and you just kind of swing it from side to side. It's a back and forth motion, so things just kind of slosh around, but in doing so, it mixes everything up. Right? And so that helps to move the chyme back and forth over the microvilli, our brush border. So we can then expose more of the brush border, the microvilli, to the chyme to maximize our absorption. I'm telling you folks, you gotta, if you don't quite understand that, all right, concept, hopefully by the end of this video here, you will. So again, we're gonna just slosh that material back and forth across the brush border so we can mix, all right, the chyme up with the glandular secretions there. And then we're going to propel, all right, the chyme, right, from the proximal end of the duodenum, right, to the distal end of the duodenum and onto the jejunum and the ileum. And we do that through the process of peristalsis. We've discussed what peristalsis is before, right? It's a series of contractual waves and relaxations, right? So the part of the intestine that's distal, okay, to the chyme, that will relax. The part of the small intestine that is proximal to the chyme will contract, pushing it distally. All right, so when we're looking here, all right, at the small intestine, and we're talking about the motility, right, this is going to occur in the intestinal phase. Remember, we talked about the cephalic phase and the gastric phase of digestion. Now we're talking about, right, the intestinal phase. So in the early part of the intestinal phase, we get segmentation. Makes sense, right? You know, we want to start to mix. We just got this chyme from the stomach. Now we want to mix it up with our own set of digestive enzymes that have been dumped into the duodenum. Okay, so we're going to mix that all up, all right, from the glandular secretions from our accessory organs, right, and the intestinal juice that resides there in the duodenum. Okay, this is governed by, hey, I've seen this term before. You guessed it, pacemaker cells, right? The pacemaker cells are going to spontaneously depolarize. And in doing so, that's going to trigger, right, the contractions of the smooth muscle, right, in the walls of the intestine, all right? And we'll see those pacemaker cells take advantage of these gap junctions. The gap junctions allow the action potentials to move from cell to cell with greater ease, less resistance. So in the late stage or phase, or the late part of the intestinal phase, that's where we're going to see peristalsis. Again, that should make sense to you because now, okay, we've mixed everything up, so let's move it on. It's time to move it on because when we move up, we move that chyme, all right, through our GI tract, we're going to expose it to more surface area, trying to get it to chemically digest even more, but more importantly, absorption. We're going to be absorbing things, all right? So we'll see, all right, motilin here is going to be the hormone that is going to help with peristalsis, okay? It's going to be the hormone that is going to stimulate peristalsis here. And so this will continue from the proximal end, all right, the duodenum, all the way down to the ileum, the distal end, right? So we can move things into the large intestine, all right? Which brings me to our next reflex, right? So we've moved our chyme through the 3 different regions of the small intestine. And it's time to get it into the large intestine. So here's another reflex. It's called the gastroileal reflex. And this is where we're going to see at the distal portion of our small intestine, right? When the ileum is contracting, right, it is going to be pushing the contents, the chyme, against the ileocecal valve. All right, inside the valve, you have the sphincter. Okay, so the stimulus is going to be the contraction of the ileum, moving the chyme, the contents there, all right, to the distal end towards the ileocecal valve. All right, and this will cause relaxation of the ileocecal sphincter. And when that occurs, the ileocecal sphincter opens up, and now our chyme can move from the ileum into the cecum. And of course, we have this wonderful hormone, CCK. Not only does it help to slow down the secretions in the stomach and the motility of the stomach, but it's going to help assist, all right, the relaxation of the ileocecal sphincter. Okay, so the valve can open up and our chyme contents can move into the cecum here. All right, so this is an amazing reflex. Trust me, this is one of the reasons why when you start to eat food, okay, not too long afterwards, for some people, they'll be like, I got to go to the bathroom. Well, that's because of the gastroileal reflex, right? The gastroileal reflex helps to advance material, okay, from the ileum to the cecum because you just ingested food that wound up in your stomach. All right, then we'll close the valve down, and of course, that's its job, right? Much like a valve in the heart is to prevent backflow of that chyme back into the small intestine. Okay, so let's talk about the accessory organs here, okay? So how do the secretions from the liver and the gallbladder and the pancreas get into our gastrointestinal tract? Well, right, we have a duct system that helps with that. So when we're dealing here, right, with our accessory organs, we've got what we call the biliary apparatus. And so the biliary apparatus is going to drain, okay, the bile from both lobes of the liver, okay, and those two ducts, all right, from each lobe of the liver are called hepatic ducts. So obviously the one that comes from the left lobe is going to be the left hepatic duct. The one that comes from the right lobe is the right hepatic duct. Well, they merge together and form the common hepatic duct. Now, the common hepatic duct, right, will then, all right, when we're not dealing with an actual digestion process right away, will move, all right, the bile, all right, into the gallbladder. All right, but now when it's time, okay, because you just had a fatty meal, we want to break down some fats, right? The gallbladder, all right, will start to contract thanks to CCK, cholecystokinin, okay? It will start to contract and it will actually push out its bile, all right, from the gallbladder through the cystic duct. And where the common hepatic duct and the cystic duct meet up, all right, and form one single duct, that's called the common bile duct. The common bile duct. And so that common bile duct will descend inferiorly towards the duodenum. I'll show you a nice picture here in a moment. Actually, let me show you right now while I have you here. Because where are you going to go? Okay, so here are the right and left hepatic ducts draining bile from the liver. Okay, and then that when the right and left hepatic duct merge together, they form the common hepatic duct. And when the cystic duct, okay, that comes off the gallbladder with the stored concentrated bile, right, where that cystic duct then meets up with the common hepatic duct, now we form the common bile duct, and that will move inferiorly down towards the duodenum here. We'll come back to the rest. A couple other structures that you should be familiar with. All right, we have, I'll show you here, two, okay, pancreatic ducts. Okay, you have the main pancreatic duct right here. All right, and then you have your accessory pancreatic duct, which is the smaller one, right? And that's going to be superior above the main pancreatic duct, right? So our accessory pancreatic duct is up here, and then the main pancreatic duct is down below, right? So the accessory pancreatic duct will contribute a small amount of the pancreatic juice, all right, that the pancreas has produced, right? It will contribute a small amount into the duodenum here. And where it attaches onto, all right, the duodenal wall, all right, that is called the minor duodenal papillae. Now, inferior to that, you're going to see, right, a larger structure, right? But we'll talk about that in a second. That's the major duodenal papillae, right? But just on the other side of the duodenal wall, you have this structure called the hepatopancreatic ampulla, right? And it is a swelling on the backside of the duodenal wall. This is where the common bile duct that came from the liver and the gallbladder meets up with the main pancreatic duct. So that's this fellow right here. This guy. That is the hepatopancreatic ampulla. It's this nice big swelling here. And then where they come together, right, they're going to meet up, right, fuse together, and they're going to then, right, secrete their secretions or eject their secretions into the duodenum, okay, through the major duodenal papillae there. So that's where we get both bile and pancreatic juices, in addition to the juices that come from the accessory pancreatic duct. And so inside that major duodenal papillae, right, there's a sphincter in there. And we call that the hepatopancreatic sphincter. Okay, so you'll see that, right, within this area here. And so that sphincter then regulates, all right, the amount of bile and pancreatic digestive enzymes that are going to be ejected here into the duodenum. So again, this is a big mixing area here, big mixing area. The duodenum plays a role there. All right, so again, the liver, largest organ, all right, on the inside of your body, largest internal organ, is going to be located in the right upper quadrant. Remember, on the right side, just below the diaphragm, right? Its job is to make bile, amongst many other things. But for the digestive portion, all right, or the digestive role of the liver, its job is to make bile. And we'll talk about what bile is used for, right? Well, here we go. We're going to talk about it right now. All right, so what is bile? What is in it? Okay, a lot of water. Bicarbonate, okay, that's great. We can use that to help buffer the acidity of the chyme. The bile salts, right, and the lecithin, both of those are primarily going to be used for the digestion of lipids. Right, we can always use some mucin. Mucin is always helpful, helps with lubrication. Right, and then of course, the pigmentation, all right, that you'll see there in the bile will help affect the color of your stool, right? And some of what contributes to the pigmentation is going to be hemolysis. When we've broken down some of those old red blood cells, right? We talked about that earlier on in the semester, when we talked about somebody that has that jaundice appearance there, okay? Usually because of the liver is playing a role in that. But let's not forget too, okay, cholesterol. When we get into metabolism, we'll talk about more of the role of cholesterol. But we know that cholesterol is found in the plasma membrane. So cholesterol is found all over the place. You just don't want to have too much of it circulating, all right, in your vascular system, in the circulatory system. But we need cholesterol for plasma membranes of the cells. We also need it, all right, for hormone production too. All right, so we have cholesterol in our bile. All right, so the gallbladder, okay, its job, stores and concentrates the bile, right? When it's stimulated to release the bile by cholecystokinin, CCK, right? That will cause the contraction, right, of our muscle layer, the middle muscularis layer there, okay? It'll cause that to contract, and then we can eject, all right, the bile out of out of the gallbladder, right? And we will cause that to move through our sphincter valve. Okay. The pancreas. Pancreas is an amazing organ, okay, because it has a dual purpose. It is an endocrine gland and it's an exocrine gland. It does both. So back in chapter 17, we were talking about the endocrine system. We learned about its endocrine function, all right, when it comes to insulin and glucagon. But the exocrine function is what we're going to focus on today, right? It is going to make the pancreatic juice here, right? That is going to help break down, all right, the contents of the chyme here. This organ is located retroperitoneal, which means it's going to be, right, posterior, right, against the posterior abdominal wall there, all right, outside, all right, of the peritoneum. And so I'll show you a picture of where it's located nicely here, right? So if you look at the pancreas, the little tip here, that is the tail of the pancreas. And if we zoom in here on the anatomical or the gross specimen here, here's the spleen, okay? That's in the left upper quadrant. So the tail of the pancreas is against the spleen. Then, right, as we move here and to the right, the pancreas starts to enlarge, right? That central portion, that's the body of the pancreas. And then this kind of large structure over here that sits, it nestles itself right in against the duodenum here. That's the head of the pancreas, right? So this structure here is going to be retroperitoneal. So will this part of the duodenum, all right? All this will be retroperitoneal. This part of the duodenum is going to be intraperitoneal. All right, so it'll be pretty far posterior in the abdominal canal, not canal, excuse me, the abdominal region there, or cavity. All right, so a little bit of a review here for the pancreas, right? When we're talking about its histology, right, we'll see, right, we have these clumping of cells. We call these cells acinar cells. They are simple cuboidal epithelial cells, right? And so we know that if you are simple cuboidal, or if you're simple epithelium, you're either going to absorb, secrete, or filter. Well, in this case, these cells secrete, okay? So we're going to clump these cells together, right? And we stick them together in these big groups called acini, all right? And so they're going to make and then release the digestive enzymes, all right, from the cells, and eventually it's going to dump into, all right, our ducts, the larger ducts, all right, the main pancreatic duct and the accessory pancreatic duct. But from these cells, all right, when we cluster them all together, they form these lobule-like structures, and from those lobules we have ducts that will eventually drain into, all right, the main pancreatic and the accessory pancreatic duct, right? But one of the important secretions here, in addition to the digestive enzymes, is going to be bicarbonate. All right, the bicarbonate is basic or alkaline, right? It helps to buffer. Remember, our pH is pretty low here, so we don't want to cause ulceration in the epithelial tissue lining the intestinal wall. So we're going to pump out, all right, this alkaline bicarbonate fluid to help to neutralize and buffer, right, the pH level there of the chyme there, so it does not cause physical damage to the lining of your digestive tract there. So what is our pancreatic juice made up of? Okay, well, we already talked about the alkaline fluid there, right? But predominantly it's water, all right? With bicarbonate, and then our digestive enzymes, we have our pancreatic amylase. That's going to help to break down starch. We saw an amylase up in the oral cavity. That was our salivary amylase, and we began starch digestion in your mouth. Okay, we're going to really pour it on here in the duodenum. We also see the pancreatic lipase. Lipase has to do with fat, so this is also going to digest triglycerides. All right, and so we saw some salivary lipase also being produced in the oral cavity. So, and also we start to break down, all right, fats in the stomach too, okay? But anyways, we're going to help to digest these triglycerides here. And then, of course, notice the term here, inactive. We've already run into proteases before, okay? Remember pepsinogen, right? We have to produce that in the inactive form so we don't damage the cell that is producing it. So we have the inactive proteases that are going to be produced, and then once they are secreted out into the lumen of our small intestine there, or the duodenum, then we can begin to continue our work on digesting the proteins there when they're activated in the lumen there. And we'll also see, okay, this is the first for this one, nucleases, okay? You probably don't remember from chapter 2, all right, when we were talking about nucleic acids, and the famous nucleic acids everyone's familiar with are going to be DNA and RNA, okay? But we are going to have these nucleases that are going to help with the digestion of any new ingested nucleic acids there. So what about this cholecystokinin? All right, we saw that the small intestine, all right, are the cells there in the small intestine, right, are going to produce this hormone, right, when chyme starts to move into the duodenum from the stomach, predominantly fatty chyme, because that is going to trigger, right, the gallbladder to contract. And as it does so, it's going to release bile. It's going to release bile. And that bile then, right, is going to help with the breakdown of fats, right? And that lecithin that's also in the bile will help to break down any fats that are in that chyme there. Right? And it also helps with the pancreas to release all that pancreatic juice and its contents, of which I just mentioned a few moments ago. So cholecystokinin does so by causing the smooth muscle to relax. Well, we saw that in the ileocecal valve. Remember we talked about that in our reflex there? All right, when we were discussing how, right, the contents of the chyme against the ileocecal valve or the sphincter there. And after you have ingested food and it ends up in your stomach, you start to produce that cholecystokinin, and that cholecystokinin it doesn't fully, but it contributes to the relaxation of the ileocecal sphincter there. Well, guess what? It also is going to contribute to the relaxation of the hepatopancreatic ampulla. And in doing so, that is going to allow bile and the pancreatic juice to flow forth, okay, from your biliary apparatus there, right, into the small intestine. And don't forget, we mentioned this in the previous video, it's going to cause inhibition, right, of the stomach's motility, all right, the gastric emptying and the gastric mixing, and the release of gastric secretion. So it's going to decrease the release of pepsinogen. It's going to decrease the formation of hydrochloric acid. It's going to slow the machinery down in your stomach. Which leads me to, since we're talking about hormones and whatnot, secretin, right? This hormone, right, is going to come from, again, the small intestine when, all right, that chyme, due to its low pH, right, secretin is going to help to release our alkaline product, bicarbonate. Where does that come from? Well, it comes from the liver there, all right, in bile, that's loaded with bicarbonate, and it also comes from the pancreas there. And its job is to neutralize the acid there that's found in the chyme. And at the same time, secretin will, in a similar fashion, help to decrease by inhibiting gastric secretions and the motility of the stomach. All right, folks, I'm going to end this video here. All right, I hope you enjoyed it. the latest installation of the digestive system by Dr. Kaz. I want each and every single one of you to have a wonderful day. Stay tuned for further videos.